Alright, ladies and gentlemen, this is going to be the next video on biology, and specifically going through and covering mutations. This is going to be the first part in a series of videos we're going to look at. Let's get started on this next video. So what is a mutation? Well, a mutation is a change in DNA sequence, and mutations can result from the DNA copying mistakes made during cell division, exposure to ionizing radiation, or exposure to chemicals called mutagens. And we can see all these different things like viruses, these mutagens, these environmental factors, going through and causing these mutations. Now, if we look at these, mutations are going to be caused by a few things. We can see that there's going to be chemicals, so household chemicals, things like cigarette smoke or abrasive cleaners. We can see things like radiation. The sun is going to emit radiation, and X-ray is going to emit radiation, which is going to go through and cause potential mutations. And then the last big category here are going to be infectious agents. Those are going to be things like viruses and some other pathogens. These can all cause changes to DNA that can cause mutations later on down the line. Now, when going through and understanding this, we need to understand for this video, there's going to be two big mutations we're looking at. We're looking at cellular mutations. Those are going to be somatic and gammatic mutations there. And then we also have chromosomal mutations. That is going to consist of deletion, duplication, inversion, and then lastly here, translocation. So we're breaking this video down into two parts. Cellular mutations, we'll look at those two, and then we'll look at chromosomal mutations. Now we're going to look at, in a second video, mutations that are directly going to go through and occur in the DNA. Those are going to be substitution mutations, which are going to result in nonsense, missense, and then lastly here, silent mutations. And then we also have insertion and deletion of individual nucleotides, which are going to cause frameshift mutations. So what I would suggest that you do is we look at these three levels of mutations. I want you to kind of write this down, really understand and get a grasp of this concept map here. And once you understand this concept map, this will give you kind of a scaffold for going through and looking at what we're going into detail. So let's go through and look at cellular mutations first in this video and understand what can happen at the cellular level. So if we look at cell mutation types here, let's go through and look at if there was a mutation to occur to a germline cell. So here are parental gametes. We're going to introduce some sort of germline mutation, and these two individuals are going to go through. We're going to see fertilization occur. The embryo is going to form, and then we're later going to get our organism. Now, in germline mutations, the entire organism is going to carry the mutation amongst all the cells because it's occurred initially at the sperm or the egg cell along the germline. This is different than somatic mutations, whereas somatic mutations, the mutation doesn't occur at the germ line, it's going to occur at the embryo. And what we see here is this is only going to cause one single area in which the organism is affected here. So if the mutation occurs at the sperm or the egg, that's going to go through and cause the entire organism to carry that mutation. However, in a somatic mutation, what we're going to see is the embryo is affected by whatever that environmental factor is, and as a result, only a patch of an affected area in that entire organism is going to be affected. So it's important for us to look at the two differences here between germline and somatic mutations. All right, so let's go through and delve into chromosomal mutations. And let's start with the first one here. A deletion is when a segment of DNA is lost, so there's a missing segment in the chromosome. These usually result in many genes missing from the chromosome. So what I've done here is I put down this diagram, and what happens in a deletion is we're going to lose a chunk of that chromosome. And if we look at this, we can label each of the parts of the chromosomes with the color there. So we have A, B, C, D, E, F, and what we can see in a deletion, we lose that orange chunk, that B. So what we're looking at here is the diagrams that we've made. The top part is representing a chromosome, and each of the different chunks of the colors is represented as parts of that chromosome. In a deletion, we are losing a chunk of that chromosome. All right, so let's look at that same diagram, but now let's look at a duplication. This is when a segment of DNA is repeated, creating a longer chromosome, where we usually see a result in multiple copies of genes along the chromosome here. So what we see in this case is we see a duplication. We see that the B section, that orange section of that chromosome, has been duplicated and it's made a longer chromosome. So now what we have is a chromosome that's going to have extra copies and extra sets of genes along that chromosome. All right, so let's go through and look at now an inversion. This is when a segment of the DNA is flipped and then reattached to the same chromosome. So let's go through. If we look at the top chromosome here, we're going to see that it's going to invert and flip. And the chromosome below it is representing what it would look like after it's been flipped. So what we see in an inversion is we see that originally we had A, B, C, D, E, and F. And then when we go through and invert that chromosome, we can see that the orange and the blue chunk of that chromosome have been inverted, where they have exchanged places. So now we have AED and CBF. We can see that in the diagram above. So an inversion is when the segment is flipped, and now we have sections of that chromosome that are no longer in the correct place. Now the last one here is going to be a translocation, and that is when two segments from a different chromosome go through and change position. So now we have two completely different chromosomes, and now they're going to be exchanging chunks with one another. Similar to crossing over, but in crossing over, it's going to exchange equal amounts here. In this case, these chromosomes aren't going to exchange equal amounts, and they're not going to be sets of alleles that we can go through and that are going to enable that organism to go through and potentially survive. So we can see we have A, B, C, D, E, and F below. We have another chromosome with GHI, JKL here. What we can see is now a section, that D section from the chromosome on the top, has been exchanged with the chromosome along the bottom. So now we have ABC, JKL instead of DEF, and we have GHI, DEF here. And in this case, we're seeing equal amounts exchanged, but we see chunks that no longer resided on that chromosome originally being exchanged amongst that chromosome. So that's really, really important for us to understand that when a translocation, it's not the same as crossing over, but the mechanism is similar. The difference is this is going to cause a mutation that can cause rippling effects along that organism and cascade effects that potentially cause the organism to no longer survive. All right, so this is going to be the end of the first video here. Did you learn? Well, did you learn a couple of things? Did you learn the basics of what a mutation is? Did you learn types of cellular mutations? And then lastly here, did you learn about the types of chromosomal mutations? This is going to be the end of the video. We'll see you all in class tomorrow.